within the biomatter, and surrounding the this core artery that is embedded within the biomatter is the ependymal cells. Okay, so these ependymal cells, as you can see, they are very tightly stitched together, okay, by tight junctions, okay, and they are also ciliated. Okay, so they don't allow, you know, just anything to pass through them. Now, the whole core of this is what we call the choroid plexus. Now, so basically, as I said, so these are the ependymal cells, and the cilia on them is what contributes into the movement of the CSF. So by beating, they contribute into the movement of CSF. Okay, so let's learn now about how CSF is secreted. Any questions so far? All good. Now let's learn more about the secretion. Okay, so as you can see here, so this is a choroid plexus, okay, which is heavily vascularized. As you can see, these arteries going in. Okay, this side here is the blood-facing side, which is called basolateral side. Okay, and on this side we have the ventricular side. Okay, which is called apical side. Okay, so if we zoom in more into this choroid plexus, this is what we will see. So we will see the epithelial cells, which are the ependymal cells, which we said are tightly stitched together, and they have cilia. Okay, and as you can see here, we can see the blood capillaries, which are fenestrated. You know what fenestrated means? What does it mean? Sorry, holes. So they allow substances to go through them. Good. Now, if we zoom in even further into just one cell, okay, so this is what we will see. So as I said, this side is the ventricular side, and this side is the blood-facing side. So how is basically the secretion of CSF regulated, or how CSF is produced? CSF consists of multiple things, as we will see in a second, which basically cations and anions. For example, movement of cations and water, so here we have an ependymal cell, as you can see here, that, as I said, that CSF is highly regulated. So we have channels and transporters on the apical as well as in the blood-facing side. Okay. So, for example, with the movement of sodium, it's basically we know that we have sodium potassium ATPase channels on the apical side, which what will they do is that they will pump sodium to the ventricular systems and recruit potassium to the ependymal cell. So this is an active transport mechanism, isn't it? Okay, and so by pumping sodium to the ventricular side, we have lower concentration of sodium within the ependymal cell, which will activate a secondary active transport of sodium from the blood. Okay. Now, also what we have a secondary active transport as well, which also contribute into the pumping of sodium, which is through the sodium potassium chloride pump. Okay, so as you can see here, that these pumps, the primary and the secondary active transporters, they do contribute into pumping sodium into the ventricular system, thus contributing into producing the constants of CSF. Now, what about potassium? We know that potassium, we have a high concentration of potassium within the ependymal cells, and so they usually, the potassium tend to move freely down its concentration gradient via a potassium channel. Okay. What about water? As you're probably familiar with this, that water tends to diffuse down its concentration gradient, okay, through channels which are called aquaporin channels. Okay. So that's with the movement of cations. What about anions? Anions like chloride, okay, is transported into the cerebrospinal fluid via the sodium-potassium chloride pump, okay? And it will be transported or recruited from the blood via the chloride bicarbonate exchangers, okay? Now with bicarbonate, which is very important for stabilizing or, buffering for maintaining a regular pH, okay, regular acidity, bicarbonate is locally made within the ependymal cell by carbon dioxide, by joining of carbon dioxide with water, okay? And this reaction will be mainly facilitated by an enzyme called carbonic anhydrase, okay? And so bicarbonate will be transported into the CSF down its concentration gradient via its channel, okay? So this just to give you an idea that whatever CSF, all the things found within the CSF are highly regulated. It's very regulated mechanism, okay? Because this is the fluid which will bathe our brain, and it has to have, it has to provide the brain with the right micro environment that it will allow the neuronal cells and, to function properly. So it's very highly regulated. Any questions so far? Okay, good. Now, is CSF different from the blood? Yes, it is. The main difference is that it lacks plasma proteins, okay? So there are no plasma proteins within CSF because these plasma proteins are gonna, produce a devastating damage to the neuronal cells, as we will see later, okay? So one of the main differences is that proteins are in very low concentration in the CSF compared to the plasma. What else? We have higher concentration of sodium, chloride, and magnesium in the CSF compared to the plasma. And we also have lower potassium and chloride in the CSF compared to the plasma. CSF is mainly water, okay? So 99% of CSF is actually water. And typically, an individual make of around 400 to 600 mils of CSF per day. With any given time, as you are sitting now, you have around 150 mils, okay? So it gets changed 3 to 4 times, okay? Per day. So that was secretion of CSF. Let's talk about circulation. I guess you guys are familiar with the circulation of CSF. Any volunteer to come up to come in the front and explain circulation? We'll clap for you. Anyone? I'll pick. Marwan, you want to give it a try? No. Okay.
Okay, so
yeah, come here.
Huh?
Yeah. Just
yeah. So as the doctor said, we have the choroid plexus located in each of the ventricles. As we said, it makes CSF. So first we start at the lateral ventricles. Choroid plexus in the lateral ventricle will secrete CSF.
What is the lateral ventricle? Show them.
Oh, okay, on the
Yeah.
So the lateral ventricle is situated here. it has a choroid plexus which will secrete CSF. Then it will flow via the intraventricular foramen to the third ventricle. In the third ventricle, we also have a choroid plexus which will add more CSF. Then through the cerebral aqueduct or aqueduct of Sylvius, it will flow into the fourth ventricle. The fourth ventricle, there will be more CSF. Then it will flow through two lateral apertures and one median apertures. As we took them in anatomy, the lateral apertures are the apertures of Laschus and the medians are of Magendie. Then it will accumulate in one of the areas here and it will pass the brain and the brains. Then for reabsorption, it will be reabsorbed at the villi.
Very good. In the top, yeah, it will travel to the top.
It will travel to the top and circulation starts here.
Excellent. Can you clap for Marwan? Our hero. Very good, Marwan. Very good.
Thank you.
Okay, so as Marwan explained very nicely, so from the lateral ventricles, it will pass, okay, go to the third ventricle where it will add more CSF, and then through the cerebral aqueduct into the fourth ventricles where it will exit through three openings, which are the foramen of Loschka and Magendie, okay, which is the lateral one, the median one, and then it will travel in the subarachnoid space. Okay, so it's gonna go in a unidirectional. So from up to down, once it exits, it will be multi-directional. Okay, then in the subarachnoid space, okay, so as you can see here, it's exiting into the subarachnoid space. Well, it will travel down. Some will go down and some will go up, where they will be exiting through these structures here, okay, which are called arachnoid villi or granulations, okay, into the superior sagittal sinus, okay. So as you can see here, so we said it will be traveling in the subarachnoid space, then it will exit into the superior sagittal sinus, which is the large vein that's located on the top of the brain here, okay, where it will be traveling, where it will be traveling back into the, heart to be pumped again for reoxygenation, as we will see. Now let's look at this, arachnoid granulations. So basically, as we said, that CSF is going to be traveling in the subarachnoid space and it will be reabsorbed or drained via this arachnoid granulation into the superior sagittal sinus. So these arachnoid granulations are finger-like protrusions that are covered with an endothelial layer, okay? They are, bulging into the dural sinus. And so CSF will move from the subarachnoid space into the sinus because of the pressure difference. There is higher pressure in the subarachnoid space than the sinus, which will drive the CSF to exit into the sinus, okay? And so these arachnoid granulations, when there is an increased pressure within the subarachnoid space, what will happen is that they will increase their cell surface to allow more CSF to exit into the dural sinus, okay? What else? Also within the arachnoid granulations, it acts like a valve. So it prevents CSF in the sinus to go back into the subarachnoid space, okay? Now, once the CSF has exit the subarachnoid space into the superior sagittal sinus, it will travel into the confluence of sinuses, then into the sigmoid sinus, then into the internal jugular vein, to going back to go to the heart to be pumped to the lungs for reoxygenation. So simply, CSF is produced from the blood, then it will exit to the to the superior sagittal vein, to be back to the heart, to be reoxygenated again, and the cycle will continue. Okay. Now you may ask, okay, so how is the exchange of the interstitial fluid with CSF occur? Okay, and it's still this is a controversial area, and there are a lot of hypotheses, etc. But one of them suggests that, as you can see here, that we have arteries that are going deep into the brain parenchyma. Surrounding these arteries, we have the pyometer, right? So the space between the pyometer and the blood artery is called perivascular space, which are also called Virchow-Robin space. And some suggest that the exchange between the interstitial fluid and CSF occurs at this space, okay? Still, this is an active area of research, and there are multiple hypotheses about how the exchange happened, okay? So why CSF is important? Why? Why CSF? What does it do? What are the functions of CSF? Cushions for the brain, very good. It provides protections against simple trauma, yes? It lights the weight. Do we know how much a brain weight? Provide the nutrition, but do we know how much the brain weight actually? Anyone guess?
2%.
In terms of kilograms? 1.5, excellent. It's actually 1.5 kilos. And the CSF makes it feels like 50 grams. Okay, so to provide buoyancy, this is what we call buoyancy, okay? Also, some suggest that it provides nutrition, so that it regulates the homeostasis of the brain, as it provides constant pressure, provides nutrition, removes waste. Also, some studies suggest that it has a role in the development of the brain. So CSF is very important. Now, the rate of secretion and absorption, it has to be very well regulated, because any disturbance in the rate of secretion and absorption would lead to high pressure, which would lead to harmful consequences. Like the normal CSF pressure is around 10 to 15 millimeter of mercury in adults, and 3 to 4 in infants, okay? CSF pressure is regulated at multiple levels, the level of secretion, circulation, and absorption. Some of the ways that have been suggested or proposed that regulate CSF is that there are evidence to suggest that there is an autonomic nervous system regulation of CSF, and this can be directly where the choroid or the ependymal cells, they do have receptors, okay, which will, for some like cholinergic neurotransmitters from the autonomic nervous system, or adrenergic, can act on these on the choroid plexus, in order to directly regulate the rate of secretion, okay? There is also the negative feedback, because you remember that we said in here that, there are different channels on the either sides of the ependymal cells, and pressure play a role. So for example, if there is a high pressure in the ventricular system, okay, there is high pressure, what will happen is that this will reduce the pressure across the ependymal cell, leading to a lesser production of CSF. Is this clear? Okay. What else? Also, there are neuropeptides, okay, which are known to also act on their respective receptors on the epithelial cell of the choroid plexus, which would lead to reduced secretion of CSF, okay? Like arginine, vasopressin, and also other peptides. Okay, any question? And so any disturbance in the production, the rate of production and reabsorption would lead, to, high accumulation of the CSF, which is a condition called hydrocephalus. And this can be either communicating or non-communicating. Communicating if the obstruction is happening outside the ventricular system, whereas it's called non-communicating when the blockage is within the ventricular system. Okay, and so when there is an accumulation of the CSF, this would lead to compression on the brain tissue, leading to damage to different brain tissues, and different environments could be manifesting. Okay. So CSF is very important when it comes to the clinic, as you will learn later on from clinicians. Like the appearance of CSF, usually it's clear. Okay, so normal CSF is clear. It has, very low protein concentrations. Number of lymphocytes is low. Okay, there are, but it's very low. Glucose is available, but to a certain level. Okay. Now, it can be used to diagnose many diseases, like in the case of bacterial infections, there would be, for example, a reduced glucose because the bacteria love glucose, okay? And so, usually CSF would look cloudy, okay? And there is reduced glucose in CSF, okay? Also, for example, in hemorrhage, you would find red blood cells, and viral infections, you would see different types of white blood cells present in CSF. So the bottom line is that CSF is very important for diagnosis of many diseases. Okay. Any questions? All good, clear? So let's talk about the blood-brain barrier. So the blood-brain barrier is just a concept. So there is no actual wall which isolate the nervous system from the rest of the body. It's a concept. And this was the blood-brain barrier, the concept or the evidence, the early evidence for its existence come from experiments that were done like 100 years ago by a scientist called Goldman. What he did is basically injected a dye in the, in rats. And what he found is that the dye stained the whole body except the nervous system, okay? Whereas, staining the central nervous system stained only the nervous system without staining the body, okay? And so this is when the concept of the blood-brain barrier or the existence barrier was, discovered. So why the nervous system has to be isolated, or the brain has to be isolated from the rest of the body? Any idea why? Why? Why it should be isolated?
Prevents maybe microorganisms from
Very good. Just to prevent microorganisms from having access to the brain. That's one thing. What else? We talked about the excitability of the neurons. How sensitive these neurons are to changes in potassium, sodium, different ions. Wouldn't it be smart just to keep them isolated? Okay, so the blood-brain barrier or the barriers basically keeps the ionic composition optimal for synaptic signaling, okay? So it keeps the microenvironment, within optimal in order for these neuronal cells to function. Also, we have these neurotransmitters, which are very valuable, okay? And so if the blood have access to these peripheral, to these neurotransmitters, we would have disturbances of the neuronal functions, right? Also, if the brain has access to the periphery changes after eating a meal, okay, and you have, you know, changes in the blood, like for example, rising of glucose, this would also disturb the brain function, isn't it? So it has to be isolated. It also, as you said, as he suggested, that prevents toxic substances, microorganisms, plasma proteins from having access to the brain. So the blood-brain barrier is very important. Now, there are multiple types of barriers of the central nervous system. The main ones which we will talk about in this lecture is the blood-brain barrier and the blood-CSF barrier. So the difference is that, as I said, it's not an actual wall. For example, the blood CSF barrier is mainly the tight junctions that are stitching the ependymal cells together, okay? So the blood CSF barrier, okay, is the tight junctions between the ependymal cells which prevent the, you know, the blood or, you know, the whatever in the blood to leak into the CSF, okay? So this is what we call the blood CSF barrier. Whereas the blood-brain barrier, it's actually the tight junctions that are formed between the endothelial cells of the blood capillaries, okay? So these capillaries are not fenestrated, okay? And so this is what we call the blood-brain barrier. Is this clear? Okay. Now, the blood-brain barrier, as I said, is formed by the tight junction. So this is a capillary, okay, a section. And so it's formed by a tight junctions, which are made up of proteins like occluding and other proteins. And so also contributing into the blood-brain barrier is the basement membrane surrounding the capillaries, and also the astrocytic food processes that are, you know, engulfing around the capillaries, okay, surrounding the basement membrane. Now, the blood-brain barrier doesn't allow, you know, all substance. This is the function of the blood-brain barrier, is to restrict certain substances from having access to the brain. And so certain things or certain substances can cross the blood-brain barrier, like gases, like oxygen, carbon dioxide, water, lipid-soluble substances, alcohol, can cross the blood-brain barrier by simple diffusion, okay? Others, they have to go through carrier-mediated transport, like glucose, okay? It has to go through, glucose transporters, like, you know, the GLUT1. And so this, you know, blood-brain barrier makes it very difficult for the pharmaceutical companies to come up with drugs that can access the blood-brain barrier to reach the brain, okay? And this is one of the challenges that facing, medicine, because antibodies don't have access to the blood-brain, don't have access through the blood-brain barrier. Many drugs, for example, even like dopamine, doesn't have access to the, to the brain. So for example, Parkinson's patients, they are given L-DOPA, which can be transported, which is a precursor for dopamine, which can be transported via an amino acid transporter to the brain, okay? And in the brain, it will be then converted or synthesized into dopamine, okay? So this is very big challenge. Now, when the blood-brain barrier gets broken for any reason, any pathological conditions, it would, produce, a devastating consequence. For example, this would lead to leakage of many of the plasma proteins. For example, hemoglobin, okay, which will basically produce oxidative reactive oxygen species, which will be activating microglial cells. We have fibrinogen, thrombin, plasmin. Plasmin, for example, will basically destroy the extracellular matrix, and this will contribute into the cell death of the neuronal cells. We have albumin, which will basically contribute into the, accumulation of the fluid like edema. Antibodies will be having access now to the brains. It's going to be a total miss, okay? And so the brain would lose its privilege, immunoreaction privilege. And so, however, although that we have the blood-brain barrier, certain structures within the brain, they do have access to the blood, but for a certain reason. And these structures are called circumventricular organs. These are found around the third and the fourth ventricles. Okay, there are 6 of them. Okay, like you can see here, the organum vasculosum, median eminence, pituitary gland, area postrema, subconjural organ, biennial, the subfornical organ. Okay, all of these structures, they do have access to the blood for a reason. So some play the role of sensory roles where they sample the blood in order to detect things like the organum vasculosum, okay, and the subfornical organ. They basically contain osmoreceptors, which they sense the changes in the salt water levels, and they contribute into our inducing our drinking behavior. Area postrema, they have receptors which detect the presence of toxins, and they are known to contribute into our vomiting behavior. The others play secretory roles, like the ones that produce hormones, etc. Like the pineal gland is known to do what? Any idea? The pineal gland? Sorry? Melatonin, very good, which is for what? For sleep cycle, very good, to induce a sleep, okay, or to regulate the sleep cycle. Also, we have others like the pituitary, which secrete hormones like the vasopressin, oxytocin, which are, as you learned in the the reproductive system and all of that. What do? They regulate milk production, several things related to pregnancy. Very good. And others like the median eminence produce or secrete proteins, secrete hormones, which regulate the release of the anterior pituitary hormone like LH and FSH. Okay. Yeah, so this is the end of the lecture. Any questions? Guys, any questions? Okay, have a nice weekend. Okay, thank you. Sorry? Yeah, let's see if they have questions. Yes. Blood CSF barrier is at the level of the ventricular system, okay? Because this is where the, at the level of, because we said CSF is produced by the, at the core, by the choroid plexus, and so by the ependymal cells. And so the tight junctions between the ependymal cells, what really contributes into the blood CSF barrier. Whereas with the blood-brain barrier is found everywhere, within the, within the nervous system, the central nervous system. Okay. Any other questions? What is the significance of the vascular space? This is the space where the exchange of the CSF and the interstitial fluid occur. This is this is a hypothesis that has been proposed. What is the difference between blood-brain barrier and blood CSF barrier? Blood CSF barrier, as I said, is formed by the tight junctions between the ependymal cells which line the ventricles. The blood-brain barrier is by the tight junctions between the endothelial cells of the tubercles. Blood vessels themselves are not fenestrated. Blood CSF barrier ependymal surrounding lies to all cerebral blood vessels. Okay. What's the green? Can you repeat slide 5? Slide 5. It's just showing the different choroid plexus receive blood from different arteries depending on the location.